The Aquatic Symbiosis Genomics Project is a collaboration between 14 international research hubs, and it's sequencing more than 1,000 species from 500 aquatic symbiotic systems to provide insights into complex ecological and evolutionary relationships. The goal is to generate annotated, gold standard reference genomes for host and symbiont. To achieve this, an initial workflow is being developed to overcome the challenges in large-scale sequencing of diverse aquatic symbiotic systems. After collection, samples are snap frozen and a detailed metadata manifest completed before being shipped by specialist cold chain couriers on dry ice. Weighted subsamples are homogenized in lysis buffer, or trizol, with biomasher and power masher, and samples are tracked using the STS and COPO system. Long-read data are generated from a low-input library protocol, followed by sequencing on the PacBio Sequel2e. For Hi-C, DNA protein complexes are cross-linked prior to sample fragmentation and DNA extraction, using the ARIMA Hi-C protocol. Libraries are sequenced on the Illumina NovaSeq. Genomes are assembled using HiCanoe or HiFiasm, purged to separate haplotypes, and MitoHiFi assembles the mitochondrion and eventual plastids. Salsa scaffolds the genomes using the Hi-C roots, and the assembled scaffolds are separated by taxa using a combination of the NCBI blast-based decontamination pipeline, the Cobalt ID workflow, and Blob Toolkit. The assembled genomes are curated using Hi-C maps and GVAL, where Hi-C contact information, coverage, and assembly gaps are used in manual curation to scaffold assemblies to chromosome level. Curated genomes are post-processed, submitted to the ENA, and annotated de novo using the ASG annotation pipeline that generates a repeat library for each species and uses RNA-seq to train gene model predictors in Breaker 2. The annotated genomes will be hosted by Ensembl, and a genome note describing the assembly published in Wellcome Open Research. So far, DNA has been extracted from 64 species, and 39 species are in the process of sequencing. There is continuing R&D in two main areas. Firstly, in the genomic separation of cobionts, where high levels of environmental contamination in aquatic samples make the separation of host and cobiont genomes trickier. And secondly, in DNA extraction, where some taxa have proven to be more difficult to extract a high yield of DNA than others.